Okay, so my name is Wan Lam, and I joined BC Cancer and UBC almost 25 years ago. And when David asked me to give pathology rounds, and asked colleagues to give me slides, and then they gave me all these slides. And then unfortunately, I realized that I can't fit a whole lot of slides in, and then I hope that I can stay on time. Anyhow, so today I'm going to talk a little bit about lung cancer molecular biology in smokers and non-smokers. So for the non-cancer people in the crowd, I guess cancer accounts for about 28% of deaths in Canada. These are Canadian Cancer Society statistics. Lung cancer accounts for about 13% of all the cancer cases, amounts to about 30,000 of people would be diagnosed, and over 20,000 people would succumb to disease per year. 5-year survival is very bad. As you can see here, circled in red, about 22%. And stage of diagnosis makes a big difference. Early detection and diagnosis at early stage really helps with survival. So these background for some of you would be very elementary, but for the general audience, I'll quickly go over this. Lung cancer is mainly divided into non-small cell and small cell, non-small cell being the majority of them. And then non-small cell lung cancer is further divided into subgroups. The main subgroups are adenocarcinoma and squamous cell carcinoma, and they're further subdivided into multiple subgroups. And I'm not going to go into the details today, on the interest of time. The squamous cell carcinoma mainly found in the upper airways, and then adenocarcinoma is further in the peripheral lung. So today I'm going to touch on a few topics, current topics in lung cancer, and these are also partly work that we do in the lab as well. So I'll talk about smokers and non-smoker, genetic and epigenetic alterations, non-coding RNAs, and then some examples in terms of immuno microenvironment, and also lung tumor microbiome. So in terms in terms of mutations, tobacco is the number one cause of lung cancer, and it's mainly more associated with small cell lung cancer and squamous subtype. But smoking is not the only cause of lung cancer. There are many other causes, environmental and occupational exposure being a major cause. Air pollution, PM 2.5 is a very topical question in terms of air pollution and lung cancer. Environmental exposure also includes, surprisingly, arsenic exposure. People think that arsenic will kill you, but at chronic exposure at low dose, it causes cancer, and especially epithelial cancer, including lung cancer. I'll touch on this as an example. Asbestos, associated with lung cancer and mesothelioma, and also radon exposure. Radon emission causes DNA damage, and this is also a cause of lung cancer in smokers and non-smokers. COPD also elevates lung cancer risk, so I'll touch on that. Not the expert on that, but I'll touch on that. Genetic susceptibility also increases risk of lung cancer. There are certain loci have been identified through familial linkage analysis and so on, and these affect both smokers and non-smokers. So in non-smokers, this becomes an issue. So there's a steady decline in the number of smokers in Canada. As a matter of fact, in developed countries, lung cancer will increasingly become the disease of former and never smokers, as fewer and fewer people smoke. And this will persist for a long time because former smokers live on for a long time. And just looking at non-smokers alone, lung cancer in non-smoker alone, it ranks as one of the higher cancer type. So this is a fairly significant burden. Some of the well-known, this is dated back more than 10 years ago, people was known about this. Never smoker and smokers have different outcomes, treatment response, and molecular biology. What I like to point out is just one thing is KRAS mutation mainly is found in smokers, and then EGFR mutations are mainly found in never smokers. And the ALK gene fusion also is found in never smokers. So these are important diagnostic markers for identifying course of treatment. So about 40% of the smokers with lung cancer actually have the KRAS mutation. So that's the largest driver mutation. And in non-smokers, about 40 to 50%, depending on the population, have mutation in EGFR. And some of these mutations actually lead to response to tyrosine kinase inhibitors. And multiple generations of these EGFR tyrosine kinase inhibitors have been developed to overcome resistance. First, ALK, of course, as you know, is due to a fusion gene and typically treated with crizotinib. In BC, in our population, this is from our own data, you can see that non-smokers and smokers are very different in terms of their mutation rate, in terms of EGFR and KRAS. Locally, molecular diagnostics for non-small cell lung cancer is done by Stephen Yip, and he gave me several slides, so I actually learned a lot. And this is all that I can fit in. So his technology, that platform that he uses, the less than ideal samples that he has to cope with, and the adaptation of new assays and technologies, flex testing and so on is demanding. Okay, so from mutation, I'll go to gene expression. So gene expression in lung cancer, you can actually tell based on gene expression pattern of the bronchial epithelium of the airway cells. You can distinguish current smokers, former smokers, and never smokers. Some of these genes are reversible. So, for example, I just take the gene on the left. In current smokers, this is the red bar is the gene expression level for this gene, and then it actually goes back to the level after smoking cessation in former smoker to be more like never smokers, and same for the gene next to it. However, there are certain genes that don't go back. For example, MUC5AC current smokers, the expression level remains fairly high in former smokers. And then a better example is the GSK3B in the middle of your screen. The current smoker, the former smokers maintain much lower level than the never smoker, even after quitting smoking. So do these irreversible changes increase lung cancer risk in former smokers? So that's one of the research topic in our lab. Now, this phenomenon also extends to non-coding RNA. And when we compare never smokers, current smokers, and former smokers, we see that some of the microRNAs, in this example, don't go back to normal in former smokers. And in fact, when you look at all the microRNAs that are in current never and former smokers, labeled color-coded here, and you can see that they have shared targets. Some of them affect the same genes, same target genes. But there's a lot of unique target genes that these microRNAs affect. So non-smokers in red, the microRNAs are listed in the middle, and the target genes are on the left. So these are unique to never smokers. So smoking, in short, smoking status affects microRNA expression in the tumors, the smoking history. And when you map these target genes to pathways, you can see certain pathways are more likely to be disrupted in current smokers versus never smokers.
One seems to be frozen for me. I don't know if he's frozen for others.
Yeah,
Yes.
this is
Yeah, across the board, I think.
Ah, okay.
Curtis has horrible Wi-Fi at BC Cancer.
Yeah.
I'll text him.
Oh, please do. Yeah, thank you.
Sorry about that. I'll continue here. All right. Can you see my screen?
We can see your screen, but if you do full screen, we can see the sidebar as well.
Yes, I'm doing full screen. Okay. Is it working?
Well, I think it probably doesn't really matter, but I can see the slide and then the sidebar and then the icons at the top. So maybe slideshow or something will get you there, but I can see clearly, and I think just continue if this is
Okay. Okay, so I'll continue. So this microRNA that is only altered in expression in current smokers targets a well-known oncogene called EZH2. And in this case, you can see that the microRNA, when microRNA 138 is lowered in expression, the patients do worse. Come on in. Yeah, thank you. So the opposite holds true because you see overexpression of the target gene when the microRNA regulation is gone, and then you see the reverse effect in terms of survival. Okay, so environmental carcinogens. I'll use arsenic as a very quick example, and I'll make up time because I'll drop some slides. So in Chile, drinking water, arsenic in drinking water is an issue. And so in northern Chile, there's an area, I don't know if you remember, several years ago, there were these miners that were trapped in the mine, 33 miners, and they had to spend a month underground. It's in that area. So we collaborate with people there that study lung cancer and in non-smokers. These people, they don't smoke, yet they get lung cancer. And long story short is that this is because of chronic exposure to arsenic in their drinking water. This is not just a problem in Chile. This is certainly a problem in North America, especially in Canada. There's in Yellowknife, there's the leftover from the Klondike. There's about 200,000 tons of arsenic trioxide just sitting there, leaching into water. And So why is this? Why does it cause cancer? Well, the way that the body gets rid of arsenic is the first step is that it requires methylation. It uses the same substrate that is being used for proper DNA methylation, S-adenosine methionine. And in doing so, it depletes methionine. It depletes the substrate for DNA methylation. And the effect is devastating. You can see that not only that the metal causes reactive oxidative species, the depletion in
your slides are not moving forward.
what's that?
Your slides are not moving forward on the feed.
Oh, okay. Can I do it this way?
It moved. We're now on the Hubo et al. Molecular Cancer 2013 in the corner.
Right. So did it move before? Was it working before?
You were a bit trapped on the microRNA, the slide you started with talking about chili.
I see. I did. That's what I did, and then when I did that wouldn't work. Okay. So I'm going to have to stay with just using this. Okay. Yeah. Thank you. Sorry about that. Okay. So because of the depletion of the substrate for DNA methylation, this caused all kinds of genetic consequences. So when we compare the genome of arsenic exposed and non-exposed tumors, you can see the difference. You can see the red marking the arsenic exposed genome. You can see all these peaks of representing frequently altered DNA alterations mapping to different places. So you can see red unique to arsenic and blue unique to non-exposed. And then when we sequence the genome, we can see that the arsenic lung tumors don't have too many mutations as compared to lung cancer from non-smokers due to other causes. And when we look at the base transitions, we see also a totally different pattern. So on the interest of time, I'm not going to go on very much longer about that. So arsenic represents environmental exposure. And then lung diseases, I guess it would be expected that chronic inflammation would have something to do with leading up to cancer as well. And we mapped several years ago, we mapped 140 genes that are highly methylated in COPD airways. So the question is that would that represent the first hit in activation of the two alleles of a tumor suppressor gene? And so we looked at that, and we looked at tumors with or without from patients with or without COPD, and we saw, based on the gene expression, we see the expected functions being disrupted. But interestingly, when we look at when we overlay the gene expression with the DNA methylation, we see some of these genes. This is just an example of the pathway. Some of them have both alleles inactivated, either by deletions, mutations, or methylation. And we do see in the orange circles where we see the tumor showing both both alleles inactivated, and going back to the airway, from the airway samples from the same individuals, we see methylation of those genes in the airway, in the COPD patients. So that's very interesting. And we're working on identifying some of these genes, and we have some leads, some cell cycle genes, and there are multiple protein-protein interaction that lead us to believe that we see some of the interesting genes. Okay, so next topic is genetic and epigenetic alteration, and oops. And so, as we know, TCGA has sequenced, NCI has sequenced more than 1,000 lung tumors in the, I can't do animation here, so, but on the left is adenocarcinoma, on the right is squamous cell carcinoma. So 1,000 tumors. The take home here is that we still see more than half of the tumors with no known identified driver genes. So there's a lot of work to be done, even though knowing that some of the tumors have mutations in KRAS and MET oncogene and so on. So a majority of them don't actually have an identified driver gene. So why is this? I think it's because the low frequency events. So for the same gene or for the same pathway, it could be hit at different places, and each one of these events could be a very low frequency event leading to some important gene and functions being overlooked. So I'm going to just give a few examples very quickly. So here's a gene called UYA4, and if you look at DNA sequences, only less than 3% of the tumors show mutation. So this would be probably a lowly prioritized gene to look at. But when you look at all the other potential ways to disrupt the gene, methylation, deletions, and so on, you see that altogether, 67% of the tumors analyzed show at least one allele being disrupted, and 20% of the tumors show biallelic inactivation. So this is something that's easily overlooked if we were just going by simple sequencing. And this gene holds true, and it impacts survival. It actually maps to a susceptibility locus, a well-known locus on chromosome 6q2325. And the function of these genes involves DNA repair and apoptosis. Another example is a gene on chromosome 1q. This is another susceptibility locus, and people still don't know if there's more than one or more genes. So we see a gene that's amplified in lung adenocarcinoma, but interestingly, not in squamous cell carcinoma. And more interestingly is that it's a well-known tumor suppressor. So people don't really pay too much attention to that because it's not really an oncogene. So it's amplified, but it's not an oncogene. But long story short is that the activity of this gene is highly cell type dependent. So L3 can act as an oncogene in some epithelial cancers and act as a tumor suppressor in other cancers. And in the case of lung adenocarcinoma, it's overexpressed in 70% of the tumors. And it shows the classical oncogene phenotypes when you manipulate it. And we think it's a very promising drug target because when we use antisense RNA, small RNAs to inhibit the expression of L3, we see this is the kind of result that we see, just very drastic inhibition. And the picture of the animal models on the right. So, and this echoes what I just said, is that when you look at lung adenocarcinoma, you can see there's a huge difference in terms of prognostic relevance as compared to when you look at lung squamous cell carcinoma, which this gene doesn't seem to be that important. Okay, so now, so I talked about different ways to inactivate a gene, and genes are being overlooked. A gene could be also overlooked because it's part of a protein complex. So you only need to disrupt one component, and it doesn't necessarily have to be the same component gene that has to be disrupted. So if you look at the histogram on the right, you can see that by different components being disrupted, the individual component never hit more than 20%. But when you tally them up, more than 63% in this cohort of 261 tumors showed an inactivation of this protein complex. And again, if you look at the bottom part of the slide, again, we see a difference in terms of which components being affected in adenocarcinoma as compared to squamous cell carcinoma. So when we look at the same complex in other tumor types, just using 3 of the components just for simplicity, you can see in ovarian cancer is the RBX1 gene that is mostly the way that the complex is inactivated. And in lung, as I showed, KEAP1 mutation in KEAP1 is quite frequent. So this is another way to find lung cancer genes is to look for components as well. So point number three is heterogeneous mechanisms in the different subtypes. So here's another example. So activation of oncogene EZH2 is a well-known oncogene in lung cancer. It's well recognized. But in non-small cell lung cancer, to the left, you can see that there's a microRNA gene. There are actually 2 genes for microRNA 101, so there are 4 alleles. So deletion of some of the alleles causes the relief in regulation of EZH2, leading to the increased activity of this oncogene. Yet in small cell lung cancer, it's mainly loss of control, mutation or deletion of the RB tumor suppressor, or the amplification of the E2F in this complex. So if you look at just one, if you just look at microRNA 101 and you're studying small cell, you might miss it as well. Okay, lastly, then I'll go on to a different topic. Lastly, is that there are also previously unannotated genes. These are unknown genes, essentially. They are there, but they are not annotated, so we don't know that they're involved in cancer. So who could these be? What are these genes? Well, non-coding RNA gene is a good candidate. So this is an example. When you look at the NCI60 human cancer panel, there are 9 malignancy represented, and you can see that almost 10% of the microRNAs have not been found in these tumor types. And why is that? Is because, well, when people compile databases of microRNAs, and when they see it one-off in one type of sample, they don't think that it's real. So it's not even included in the prominent databases like MIRBase. So here's an example about tissue specificity. This is just switched from lung. So this picture is from liver, just normal liver. And you can see the microRNA, these novel microRNAs that has not been recognized are tissue specific and ignored. So liver is in green, the green balls represent liver, and the pink balls amongst them are bile duct samples. So they came from the same origin, and they have the same cell type, and they have the same microRNAs that were not recognized. So I'll come back to this. So going back to lung, in lung, the orange colors represent lung. This is just normal lung tissue. And you can see that the expression of these novel microRNAs group separate from other tissue types. And when we look at tumors, so when we look at tumors and normal tissues and also fetal lung tissue, we found 44 novel microRNAs. And on the interest of time, I'm not going to go into details. And they can clearly be delineated. These tissue types can clearly be delineated based on the microRNAs. And these novel genes may have biological relevance. We can see that they show differences in terms of survival. And so along the same line, in terms of these novel genes, we also see them in malignant pleural mesothelioma, and we found 154 new ones. And so, you know, are they so specific that we can use them to distinguish tumors that are lung adenocarcinoma that had metastasized to the pleura? So we would put that to a test. And these microRNAs can certainly distinguish the different tumor types. And we narrow down to a group of about 40 microRNAs that can distinguish NPMs from adenocarcinoma. And of the 87 NPM cases that we looked at, 86 were called correctly by these classifier microRNAs, and the one that didn't work, we went back and looked at the path report, and it was actually one that was problematic and was not confirmed. So this is promising in terms of tissue-specific markers. Okay, so along the line of non-coding RNA, I'm going to talk about reactivated microRNAs from fetal tissue. So we know that developmental genes are often involved in cancer. So we're looking for fetal genes that they're not expressed in adult lung and reactivated in tumors. So these are ideal therapeutic targets because they are not expressed in adult lung, right? So we worked with Wendy Robinson at Children's Hospital, and we looked at fetal lung tissue and looked at their expression and then compared to tumors and non-malignant tissues from the same tumors. And as you can see, the blue arrow on top indicates a small number of fetal samples we were able to obtain compared to the hundreds of tumors on the left and right, and so on the right, and on the left are mainly where the on the right, the red is where mainly the non-tumor tissue samples are, and then the orange are more or less mixed in with the fetal samples. So now, so the question is that are fetal microRNAs critical regulators of lung development and tumorigenesis? And some of these microRNAs map to well-known pathways involved in cancer. So we mapped the 37 putative oncofetal. We call these reactivated microRNAs, oncofetal microRNAs. So we identified 37 of them that are reactivated at a high confidence. And then it turns out that the 8 of these microRNAs target the same gene, this gene called nuclear effector 1b. And what is nuclear effector 1b? So we confirmed this by experiment by altering the binding, mutating the binding site to make sure that the microRNA are binding the right things and so on. And so this looks real. So what is NFIB? Is a transcription factor. It's essential for lung development. And And this is reflected in the functional role in regulating lung differentiation. And low expression of NFIB is significantly associated with aggressiveness in what we looked at. And I apologize for this slide. The bottom, the x-axis is a different scale. So if you look at the dotted line, that's where the 5-year survival mark is. So you can see that it doesn't do very much for squamous, but you can see in the adeno, 2 cohorts of adenocarcinoma, you can see that it has prognostic significance. And when we look at tumors from tumors that have high expression compared to low expression of NFIB, we look at other genes in these tumor samples and look at the gene expression profile. What are the prominent things that we see? We see genes in lung development that are altered. So this fits into the whole story. And so the next topic is non-coding RNA. So microRNA and short non-coding RNA is one type. Non-coding RNA is really divided into long and short. So long non-coding RNAs produce all these interesting ways to regulate genes, including antisense RNA. There are pseudogenes that serve as sponges that soak up microRNAs to relieve regulation. And so, on the interest of time, because I lost some time, I'm not going to talk a lot about this. This is one example. This gene MGA1, the antisense, is expressed 18 times in tumors, and as you can see in the histogram, and it has and it downregulates the gene. And so these long non-coding RNA are very stable, so they're very useful in terms of developing blood-based diagnostics. So the protein half-life is in the ballpark of half an hour to 2 hours, and these long non-coding RNAs, MALAT1 is a well-known one in lung. The half-life is 16 hours. Okay, moving along. Tumor microenvironment. So I'll just give 2 examples. One is, again, staying along the theme of non-coding RNA. So we look for immune-related microRNA. So we compared long non-coding RNA profiles from microdissected tumor cells to bulk tumor RNA profiles. And so the microdissected ones should have very little stromal cells and very little immune cells. So we ended up with a crude list, and that's our starting point. And then subsequently, we analyzed single cell RNA sequences from lung tumor data, and from sorted immune cells, so that we can compare and see which ones belong to which types of immune cells. And keep in mind, these are changes that we see in tumors, immune cell changes in tumors. So one quick example is that we identified this long non-coding RNA, AC00875.0.1. They don't have names, they just have numbers. It actually works coordinately with a gene in NK cells, in NKG7, this cell, and functional analysis confirmed that. So we're seeing we're seeing immune-specific genes in the tumor microenvironment being altered. So the byproduct of this is an atlas of cancer cell extrinsic immune cell expressed long non-coding RNA. Okay, I'm running low on time, so I'm just going to touch on a couple more things. And so finishing the lung immune lung tumor immune microenvironment, to finish this thought, we also work with Callum Macaulay and Marcel Guillot of our department, and they developed these cell sociology platforms that you can do multiplex IHC and then compile that information to look for cell neighbor analysis. You can actually find a tumor cell, what is its neighbor and what is this neighbor. So this is important when it comes to immunotherapy, and also for tracing clonal evolution. And in short, by looking at cell neighbors, we get a lot more information than just looking at cell density alone. And we were able to identify situations where we can predict recurrence, cancer recurrence. I'm going to move on to the last topic. I think I can finish in time. Is lung tumor microbiome. So it was a surprise to me when I first read that microbes exist inside solid tumors. And this has been seen in about 30 different tumor types, and people see that they exist. In lung tumors, but nobody knows what they do. And so most people have heard of gut microbiome, and they cause metabolic issues, and the gut microbiome is also involved in metabolizing prodrugs to make drugs to be active in cancer treatment. So, but in the tumors, there are actually microbes, not in high quantities, but there are microbes, and these microbial taxa identified by sequencing of ribosomal RNA genes, we found that they actually are the bacterial taxa that can produce essential amino acids and nutrients that the tumor cells need. For example, I'll use L-methionine as an example. Humans cannot make essential amino acids, so humans cannot make methionine. So it appears that the bacteria in the lung adenocarcinoma tumor immediate microenvironment can produce L-methionine, and correspondingly, the tumor cells express the transporter for this amino acid produced by the bacteria. And L-methionine is essential for maintaining DNA methylation in cells. So this is ongoing work. And I'll end with this one topic, airway microbiome. So our microbiome we see in tumors, you find them in airways. Typically, you think of infection when you see microbes in the airways. But when we look at bronchial exfoliate cells, brushing essentially from bronchoscopy, and sequence them, you can actually describe the microbiome. And when you look at 100 samples, you can tell the variation and the way that the samples are collected after rinsing and then brushing and so on. The variation is not that high, at least enough, consistent enough that we can do comparisons. So when we compare airway microbiome from participants in this study, there were about 200 or so from a lung health study, and we're able to come up with profiles, airway microbiome profiles. So in this cohort that I'm showing here, this cohort has about 50. They include individuals with lung cancer, meaning the red dots, and then individuals with no cancer, gray dots, most of them don't. And then the interesting ones are the ones with incident cancer. And these are the ones that don't have cancer. They're individuals with no cancer diagnosis by the time by CT at the time of sampling. So before and then develop cancer during follow-up. So after CT cleared of the person saying that you don't have cancer, and then months later they develop cancer. So this could be interesting. So we look at the microbiome composition and ask the question whether there's a shift in microbiome in advance of clinical diagnosis of lung cancer. And the answer in a small cohort, the answer is that we do see that. And in this cohort of about less than 50, we see 4 out of 5 patients with incident cancer with very high microbiome predictive score. So based on the composition, we derived a way to a test score system. So these the ones with high test score, the 4 out of 5, in fact develop cancer within 1.5 years, 1.29 years. And there's one that took longer, and that one developed cancer much later, 4.5 years later. So this is enough preliminary data to prompt us to continue this research. So we have ongoing projects looking at the timing of the shift in microbiomes in the airways in advance of lung cancer development. So almost out of time. So sorry about the rush, and I skipped over some of the details. But I guess today I hope that I give you an update about our research and touch on these topics. And I'm free to answer any questions. I have time if anyone is interested. Thank you.
Well, thank you, Wan. And I'm sorry that there